For this presentation, I will be talking about the anatomy and enzymology of fat absorption. Fat absorption first starts with the mechanical breakdown of the food in the mouth. So on the first slide, we're going to talk about the different types of fats that we are absorbing. You can see we have pizza, donuts, hamburgers, peanuts, all of which have examples of the several different kinds of fats that we intake. These fats include saturated fats, which have a hydrogen bound to every single carbon bonded on the chain, trans fats, which have a trans orientation as opposed to a cis orientation, unsaturated fats, which may be either mono, meaning there is one double carbon bond, or polyunsaturated, meaning there are two or more double carbon bonds, as well as omega-3 fatty acids, which we typically associate with eating when we eat seafood or fish. So the beginning of digestion of fat starts in the buccal cavity or the mouth. As we see here, there's a picture of a woman eating a piece of pizza. Although there is no lipase present in the mouth that comes out of the salivary glands, unlike the amylase, which breaks down sugar, no fat is actually digested in the buccal cavity. However, this is where a very important part called mastication takes place, or chewing of the food. Mastication mechanically breaks down the food so that the bolus can be then transferred from the buccal cavity to the esophagus. As we'll see in the next slide, the bolus of food will travel from the buccal cavity through the upper esophageal sphincter, where peristalsis will cause it to move down the esophagus and eventually into the stomach. Once the food enters the stomach, there is very little breakdown and absorption. There is some gastric lipase present in the stomach, but the highly acidic pH and little bit of gastric lipase causes very little absorption and very little digestion of fats. In this diagram here, we can see where the main digestion of fats actually takes place. Just to repeat, as we learned in the beginning, there's no digestion in the buccal cavity, very little hydrolysis of fats in the stomach, which are into monoglycerides and fatty acids. However, the vast majority of fat is hydrolyzed and digested and absorbed in the small intestine. We see that bile will combine with fat to create micelles, which are small droplets of fat, increasing the surface area available for digestion. And we have a complete list here of the different types of fat molecules and the pancreatic lipase and intestinal lipases that break them down. The small intestine is primarily responsible, as I said, for fat absorption, and in the small intestine, bile coats the fats to form micelles or droplets called emulsification. The liver makes the bile, which is then stored in the pancreas, and bile along with pancreatic lipase is secreted into the small intestine so that fats may be broken down into small droplets and thus absorbed. As we can see in the diagram here, the fats begin as large droplets that are then emulsified by bile salts and created smaller micelles. These micelles will then transfer through the apical membrane of the small intestine, where they will go to the smooth ER to be packaged with triglycerides, cholesterol, and proteins. From there, they will combine, as we can see, they combine with cholesterol, and the Golgi will package these into chylomicrons that then enter lacteals and thus enter into the lymphatic system. As we can see in this diagram here, fats must undergo exocytosis to leave the, not exocytosis rather, they must undergo transport to leave the small intestinal cells that will actually transport them to the interstitial fluid, where they will meet the lacteal and then go through to the lymph to the vena cava. The fats enter the apical membrane of the small intestinal cells via diffusion. They're able to diffuse easily across because of the bile salts that coat them. This is a rather simple and quick breakdown of the enzymology and absorption of fats. However, I feel that it is comprehensive enough to understand the true breakdown and absorption of fats in our body. This concludes my lecture. Thank you very much.